Hi everyone, welcome back to the channel. Today we are going to cover a study guide for BOT2313 Module 4. Let's get started. Okay, so we have 3 units or chapters under this Module 4. They are Chapter 8, Plant Genetics, Chapter 27, Plant Hormones, and Chapter 29, Soil and Plant Nutrition. So let's get started with Chapter 8, Plant Genetics. Okay, so in this chapter, so basically we start with basic Mendelian genetics. So the best way to really study this chapter is start with definitions, the vocabulary, what is crossing over, diploid, dominant, recessive, gamete, recombination, genotype, phenotype, heredity, homozygous, heterozygous, P1 generation, F1, P generation, F1, F2 generations, and then basic crossings, right? So we have here a little chart basically showing the dominant versus recessive. So one of the very first experiments of Mendel, which he did with his pea plants. So in his experiments, so he crossed purple flower pea plants with white flower pea plants, and in F1, so he got all purple. And then when he self F1 generation, he got 3 purple to 1 white distribution. So that shows basic one single dominant gene. And if there are two dominant genes, then the ratio goes up to 9:3:3:1. And then we need to use probably a Punnett square to actually easily sort out and show the results of these genetic crosses. So if when we get 2 different alleles, they will be heterozygous. If the 2 alleles are same, they are homozygous. Phenotype always equals genotypes times environment. Basically, genotype is the genetic makeup, but the environment is non-genetic part like nutrition or temperature or light availability type of other conditions. Okay, so and genetics also use probability. So like what is the probability of having a plant with white flowers? You can calculate that using probability. As we know, these are rules that actually Gregor Mendel found and discovered in his pea plants, and he published 2 papers in his time in 1856, and then today we are actually he's considered the father of genetics. So next chapter is plant hormones. So plant hormones, they are also called phytohormones. Basically, there are auxin, cytokinin, gibberellins, abscisic acid, and these are the big ones you see here. They all have an impact on gene expression, as we know, but they might have impacts on different things, positive or sometimes negative impact. Right. So a little more information about the auxins and cytokinins, especially 2 large ones. So there are synthetic ones as well as the naturally occurring auxin or cytokinins. Basically, when you're studying it, make sure that you know their structure and you know where they are biosynthesized, where they are transported, and what impact or effect they do, right? And then also another important is auxin-cytokinin ratio, right? Especially in plant growth. Okay, so gibberellins is the next big hormone. GA3 is one of the most well-known gibberellins. They are mostly producing shoot apical meristem and root apical meristem. Ethylene is only gaseous hormone. Abscisic acid is we see in mature leaves, also fruits, and especially in seeds causing dormancy. There are some newly established plant hormones like salicylic acid, brassiloids, and JAs or jasmanic acid. So make sure that you also review those too. And our next chapter is basically plant nutrition and soils. So this chapter is very important because actually, as we know, most plants are grown in the soil, and they get their nutrition, most of their nutrition from the soil, right? So there are several different types of soils, but some are more fertile than others, like loams type of soils. Most fertile, is it considered most fertile or best soil in the world? We have 17 plant nutrients, and if the plant is missing any of these nutrients, it shows the signs of nutrient deficiency. You can divide plants in 3 parts: young part, middle part, and older part of the plant, and then this deficiency might show up in one of these 3 places. Upper leaves, usually like nitrogen, we might see some light green color in the nitrogen, but the zinc we see in more older leaves. Phosphorus is also an old leaf, and magnesium is more old leaf. But iron and calcium definitely are young leaf deficiencies of nutrients. So we divide these nutrients in 2 groups: macro and micro. Micro also called trace elements. Trace elements like chloride, iron, magnesium, boron, copper, molybdenum are some examples. So this chapter also talk about soil types, like loam soil I mentioned, like very fertile soils, but the sandy soils are less fertile because they cannot hold the nutrients very well. Silts and peats and clays are some other types of soils. So 3 major plant food that actually commercially most common is this NPK, nitrogen, phosphorus, potassium. As we know, nitrogen is very big one. Basically, it's involving chlorophyll production, vegetative growth, and healthy foliage. Phosphorus involving flowering DNA synthesis, and potassium is involving, again, healthy growth and synthesis of proteins and carbohydrates. So this, make sure that you know all the macro and micronutrients, their symptoms, and the soil types, and then also some bacteria like rhizobium, which helps certain plant types, or some fungi like Mygarrhizae helps a lot of the plant species to get certain nutrients. So this is the review of the 3 chapters in this module 4. Thanks for watching, and feel free again to email me or connect or subscribe to this channel, and reach out, and good luck. See you in next video.